Hi, I'm Laura Rassenti, and I see some familiar faces, but just in case you don't know me or you haven't heard me speak before, I worked for Dr. Kipp for 28 years now. I basically started his lab at Scripps before, and then we moved on campus, and then we moved here when the Morse Cancer Center opened several years ago. And my background is in molecular genetics. I'm not MD, I'm a PhD in basic molecular genetics, and I got my PhD at the University of Arizona, and then I worked as a postdoc at Stanford University for 4 years, and that's where I met Dr. Kipp. He was working on his residency there, and then he moved to San Diego, and a year later I moved here because of my husband and other things, and then I started working in his lab, and I've been here ever since. So I have a long history, and you look up our names, and we publish papers on CLL, I'm sure you know. So today, Sheila asked me that to give a talk on why is it that people that have mutated IgVH genes, they do better than people that have unmutated immunoglobulin VH genes? Because she said you are interested to understand why this mutation in this case is good, because you hear about nowadays everybody's sequencing the whole genome, sequence this and sequence that, and they find all these mutations with breast cancer genes and prostate genes, and and mutation is supposed to be bad for you. Well, mutation is sometimes good. You need to have mutated genes, and I will explain to you why in this case for these B cells, they are really better B cells than the B cells that do not have these particular VH genes that are involved in this leukemia. So I'm going to be showing, I will explain to you in a minute, this is an antibody, and you're going to see this picture several times throughout my talk. And as I said, I'm going to talk to some of the people in CLL have particular region of this immunoglobulin gene with mutations. Each of these little dots or arrows is a mutation in that each line is a sequence of a gene, and each dot is a mutation. So you can see the people on the top have many mutations in this particular gene, and the people on the bottom, you don't see any little dots, so that means they do not have any mutations in that gene. So I will explain to you how we do this and what this means in a minute throughout my talk. So first of all, the reason that, as you know, and this is a kind of like a survival curve, or where the probability of a patient to be treated, and we do many of these in the lab, and this is the time that it takes those particular patients to be treated. And there's many publications that say that if you have this unmutated VH, no mutation in this particular sequence, you go to treatment much sooner, because let's say 50% of the people get treated within, several months or a year from diagnosis, and instead if you have mutated in this particular gene, you go to treatment later. But they will eventually get treated, but not as soon as these people. And this is kind of where it all started, and we try to understand the biology. Why do these people do better than the unmutated people? So first I have to give you a little bit of biology lesson about CLL cells and how we decide whether a gene is mutated or not, this particular gene. And if you look under a microscope, this is what a CLL cells look like. They have a very big nucleus and a small cytoplasm. And Dr. Kipp always uses this slide, and I think it's very descriptive. If you look at them even closer in a very, very specific microscope, obviously, we see that these cells are not just plain cells with nothing on the surface. Like any other cell, they have many markers, many molecules that we give them certain numbers. CD means cluster differentiation, and it's an old nomenclature. Tell me something.
These lymphocytes or whatever?
These are lymphocytes. These are CLL leukemia B cells.
Okay, B cells.
Lymphocytes, right. And I'm going to show you how we isolate them from your blood in a minute. But I'm just giving you a background on that. There's many surface proteins on these cells, and they're given a number according to when they were discovered. So, CD20, we have some antibodies against some of these. CD40 for gene therapy, Dr. Kipp has done many studies on those, and so on. And the main cells and of course, it's kind of like the phenotype of a cell. It's like a person has blonde hair or red hair or white hair or light brown hair. Depending how much and which protein you have on your surface, it identifies the type of cells and where in the development of these cells you are as a B cells in the immune system. And today I'm going to be talking about the VH special protein, and that you guys want to know about the mutation side. So this is also a protein on the B cells, and it's also on normal B cells. It's just that normal B cells, we have different amounts of these proteins. But what's particular, and that is very particular about the CLL B cells, is that they have both CD19 and CD5 on their surface. So a pathologist, and again, I'm not a pathologist, but when a pathologist stains the cells from your blood and they see that they're co-expressing both CD19 and CD5, that's the first indication that the CLL B cells. Because usually normal B cells only express CD19 on their surface, this particular molecule. And CD5 is usually found on T cells. And the immune system is made up of B and T cells that interact with each other. And for some reason, because of all the changes that happens to these cells, they happen to express the CD5 protein, which kind of identifies them as the CLL B cells. So, of course, if you look at these in a 3D effect, this is just a cartoon that these protein, these could be CD5, they have different shapes, right? Because the protein is made up of amino acids and it folds and it has a certain architecture on the top of it. So it's kind of a really neat slide because it shows you, and these are just a few. I mean, a cell, a CLL cell, like T cells, and they have thousands of these proteins that are sticking out in their environment, waiting to get bound by some other protein and do all the interaction that I'm going to be talking about. And some of these proteins are important for the growth and survival, and the cell adapts to turn on off certain genes that make these particular proteins on their surface that makes the cell survive or die in its environment, for example, in the blood. And so Dr. Kipps has written an article, and it's very important kind of picture here, that says the CLL cells is not like alone in the blood. It's really surrounded by many other very important cells, and it's the neighborhood of the CLL cells that also determine what kind of cell that is, whether it can survive in the blood for many years, be resistant to treatment, be going to apoptosis or not going. So the CLL cell is not living in a vacuum. It's a very complicated system, and our laboratory works on the immunoglobulin gene, which I'm going to talk to about. It's this little protein right here. It works on CD40. It works on nurse-like cells, and you've probably heard some. You can look up papers on these. There's dendritic cell. There's stroma cell. All of these cells find a way to keep each other alive in the blood through selection and or die. And some of the treatments, and I can't go into treatments today, but like rituximab or lenalidomide, kind of with trying to disrupt some of these interactions. For example, from CLL cells to the nurse-like cells, even though let's say you can't really kill the CLL B cells, but if you disrupt and put agents in chemotherapy that disrupt some of these interactions, then the CLL cells can't get like survival signal from nurse-like cells, and the CLL can die. So there's a lot of cross-reaction and talking between the cells, and us as biologists, we have to understand kind of the whole neighborhood of the CLL B cells, because it doesn't live there in the blood all by itself. So I'm going to be talking about this little circular thing right now, and you can see the situation is much more complicated, and it's not just that one thing. Yes?
How long have they known about all of those?
The microenvironment, we wrote a paper with Dr. Jan Berger from MD Anderson Cancer Center, I would say like 10 years ago, 8 years ago. I mean, it's not like 50 years.
Yeah.
A lot of this stuff is within the last 10 years. And the field is now with the genetic, all these machines and kits from this company, it's going faster and faster. So we're learning more and more, like every year there's new publications that new proteins are found and they interact. And so it's pretty recent, yeah. Okay, so first of all, I'm just going to give you a little background on the biorepository. I work also in the CLL Research Consortium, and a lot of your samples, if you sign the consent, and I'm sure Sheila has explained to you, are put in the consortium because it helps us get large volumes of CLL patients to make like statistical analysis and understand the biology of many patients. And the CLL Consortium is again headed by Dr. Kipps. And the consortium, as you know, is a group formed to undertake an enterprise that's beyond the resource of any one member. So not any one of these scientists are in part in the consortium for Dr. Kipps really work alone, but they work better as a group. So we help each other out a lot. And a lot of the VH sequences and all of these things that we found are by putting many patients' information together. There's more patients coming. So hi. The consortium is made from several research projects, and I had the tissue core, which is what I'm going to talk about today. And there's investigators that are specialists in each of these molecular, biochemistry, immunotherapy, or pharmacology that actually come from different parts of the country. And you see UCSD is the headquarters, and we're right here. MD Anderson Cancer Center is the main another one that's part of the consortium. And this is Jan Berger at MD Anderson that I was talking to you about, kind of discovered the nurse-like cell interaction with Dr. Bill Weirda over here. And many of you maybe have heard of these other CLL doctors that are pretty famous in the world about curing CLL. And Mayo Clinic is up here. We work with them. Ohio, North Shore, Dr. Kanti Rai at LIJ, who's like the father of CLL. He devised the Rai classification system that all of you hear, Rai stage 1, Rai stage 2. Dr. Rai is, we work with him very closely. And there is the Dana-Farber at Harvard with Donna Newbert, the statistician. And now we have also a lot of sequencing going on at the Broad Institute with Dana-Farber. And of course, Dr. Gribben, who used to be at Dana-Farber, now moved to London. So we work very closely with Dr. Gribben too. So just to show you that there's many people involved in a lot of these research for CLL that I'm going to show you. Now, the tissue core, as you know, I received your cells. We process them and store them in, not just from here, but also from these other CLL people across the country. And then we basically have a biorepository. Now, up to date, we have over 6,000 CLL samples. And for all of these samples, we do a lot of tests to characterize it, to understand the biology. And the main one that I'm going to keep talking to you about is this VH. But don't forget, we also we have cytogenetics that you guys give us, that you do a karyotype from Sheila's, the local people here. We do, we need whether the demographic, the age, or diagnosis. We do a lot of plasma to do serum and ELISA samples, also with MD Anderson, and so on. And the very important samples that we also use are serial, longitudinal, because we want to see how your disease changes with time, how things change as your disease progresses or stays stable. So these are very, very important serial samples. So what do we do with your blood? And this is going to show you how we get to the mutation status. So we take the blood, and we have a technician that all she does for me is process your blood samples and freezes them away. With them, we do the ZAP70 that you've heard about. We make DNA, which is what we use to do the VH status, and we also store the extra cells for future studies. Now with the blood, when we spin it, we also use the plasma, the liquid part, the part that doesn't have the cells, and this is done for ELISAs, for antibodies, and other very important studies. Or you've heard Sheila give you that little buccal mucosa swabs that we kind of like are a hassle to do, but those are important also. And we look at the germline DNA, the DNA that's not the tumor DNA, because the tumor DNA is in the blood, for genetic studies. Now, this is what the blood looks like after we spin it. The plasma is right here. Your lymphocyte mononuclear cells are these white layer. That's why they're called white blood cells, WBCs that you see. And this is where the technician puts a pipette in here, and she goes in and basically sucks out this middle flat layer here. And that's what we put away in the freezer to do DNA, and that's what we study. Those are leukemia, the B cells, the white lymphocyte mononuclear, one nuclear cells are here in the middle. And then these are the red blood cells, which we don't do red blood cell studies that we discard. So when you see the word PBMCs, it means peripheral blood mononuclear cells, and they're right there in the middle. And of course, if a patient has a high white blood count, this layer is bigger, and it really, literally, we can spin and we can see how many white blood cells a patient, kind of rough eyeballing, how thick that layer is. It's like, advanced or less advanced disease, but it's just kind of looking at, that's me a lot. So this is the blood, and what we do is once we freeze those middle layers, we have 7 huge liquid nitrogen tanks downstairs with lots of racks like this, where we freeze, and I have the technician put them in specific spots with the patient identifier, all confidential, that we can go there and pull them back out and then do DNA and RNA, which is what I'm going to show you right now. So what's in, as you know, DNA is in the nucleus, and then RNA is made from DNA, and then it gets processed and then becomes either non-coding RNA or messenger RNA, and it makes a protein. So we have to start from the DNA for the VH studies. And what we do is, once we have the DNA, we do, and now you've probably heard of this, is a very famous technique, PCR, polymerase chain reaction, which was established, and the person got a Nobel Prize many years ago for inventing this reaction, which is now used in every probably molecular lab in the world. And what we do is, we have a DNA sequence, a DNA in general. And you say, oh my gosh, you've got thousands of genes in your body. Well, how do you know you pick up that one VH? Well, what we do is we know this sequence and we make little primers, little complementary, the DNA is ATCG, right? So A binds with a complementary base. So we make a primer that we know it's going to stick only to that particular identical sequence that goes, this is the sense primer, and then the primer that goes on the opposite way is called antisense. And it binds here because A binds the T, a G binds the C. And it's going to go find them and stick to here. And then we open up the DNA. Then we denature, and the primers are neo here. Oops, sorry. The primers are neo to the right sequence, and the PCR reaction, which I can't go into too many detail, has an enzyme that's called Taq polymerase. And it means thermo aquaticus, and believe it or not, it was found in the hot vents in Yellowstone. I don't know if you know the story. The guy got a Nobel Prize. Because this enzyme, it's a polymerase. That means it copies the DNA. But the problem is, he had to invent this technique because this DNA has to be denatured, right? You got to open up the DNA for those little primers to go stick to them, because it's not going to stick to it if it's a closed zipper. It has to open it up. So to open it up, we've got to heat up the DNA to very high temperatures, like 94 degrees, over and over again. And most polymerases, that's pretty hot for an enzyme. It's going to get denatured. But this person found this enzyme in the heat vents, in the Yellowstone, boiling water, and the enzyme works in such a, so he isolated and cloned it and so on. And now they sell it like, it's very cheap. It's been going on for a year. So we use this enzyme, Taq polymerase, to amplify, because it goes there, and once that primers are attached, it goes in like a little, kind of like a train on a railroad track, goes, it makes a copy. And this happens over and over again. We do many cycles in a little PCR machine, and it takes a couple of hours. It's not very long. So we've got all the people's DNA in them. We can do like 20, 30 at a time. And we amplify. So from two bands after once, and now we have 4, and they're identical. So and then 4 another cycles time, then it becomes 8, and then it becomes, so it's exponential, and we have a ton of DNA, which we can look at on the gel. We can actually, and you see from a target sequence that we start from the DNA, the primer anneals and makes new DNA. Well, then this opens up again, it's hot. It makes more DNA. So after many cycles, and 30, 40 cycles, we have a lot of DNA from these 2 little strands. Now we have very many, and we can figure out what this DNA, VH, we can sequence it now, because we have a lot of it. You can't just sequence 2 strands of DNA. You don't have enough in a little tube, right? So what we do is, for example, it's just another example. We take the cells, like I said, we make the DNA or RNA extraction, and then we PCR, and then we take this sequence, this little, it looks like water, right? You can't really see it. We send it to a sequencing lab in town, and it's really cheap. It's like $9 to sequence this little, it's only 200, 300 base pair, don't forget. It's not the whole genome, it's little. And in the lab the next day, because it's very automated, they send us something looking like this, okay? And then we put this in a database. It's not that complicated. And the database says whether there's whether it matches or not. So for example, here's your DNA, and it just looks like water. So the DNA has a sequence, A, T, C, G, which is random, depending what gene you're looking at. And then each of these, if one of these is mutated, A binds with C, with T, a T with A, and so on, that's the genetic code. If there's a mutation, for example here, there's a G that became a C, then we're going to see, like in your final, this is not really the best example, but we don't see that C anymore. It changes into a T, let's say, and we can actually get a readout from the sequencing lab. But usually, if the genetic code, just so that you know, every 3 bases becomes an amino acid, right? And an amino acid is then put together to make a protein. So if there's a mutation, let's say here, the U, it's uracil for RNA, the G is changed to a C, it's not going to be a methionine amino acid. It might be an isoleucine because the genetic code is 3 bases will make a certain amino acid. If one of these 3 bases is mutated, the G becomes a C. That codes for a different amino acid. Just the rule of life, the genetic code that was invented, that was discovered. So different, of course, if there's a mutation here, then you're going to get a different amino acid. And if you know, amino acids all come together into a protein. And they don't stay like a string. The protein folds in a 3-dimensional structure. It's very complicated. I don't know. There's binding force and each amino acid has a certain structure which then binds in each other. And it'll make a structure. And the protein, I always tell people it's like in this situation, like a baseball glove. If it's folded in a certain way, a certain other protein can bind in it, like the baseball ball can bind to it. If it's made in a more pointy way because the amino acid is different, so the shape is different because there was a mutation, then a different type of ball is going to bind in your baseball glove, right? It's not going to be round, it might be pointy. That's what fits more. Because I want you to understand this because I'm going to be talking about antigens and antibodies, and it's all about how the antigen binds to your antibody, which is your baseball glove. So do you understand so far? I mean, it's pretty straightforward. You just have to envision a 3D effect, this thing sticking up on front of your cell, and it's ready there to bind something that's in the environment that can fit in that groove or not. So it's important what fits in that groove. So now that you have a little better understanding of the background, we're going to go into the biomarkers and prognosis for CLL. Now, as you know, we look at many biomarkers to determine whether a person has a probability. It's not like a done deal because there's exceptions. A probability of having a bad prognostic or good prognostic in your cell. And we look at several things as doctors or biologists. We look at the lymphocyte doubling time, the serum micromarkers, and of course the VH mutation status, which is what I'm talking about now. And you've seen ZAP70, CD38 is also sometimes good or bad prognosis. And also very important is FISH cytogenetics abnormalities. If you have a 17p deletion in your chromosomes, it's not a very good sign. Yes?
If you have all of these prognostic markers
right.
that are negative for you, does that give you a worse prognosis than if you have 3 good and 3 negative? Or can you say that or not?
I mean, it's not good. Yeah, but there's always exception to the rules, and it depends which 2 you have. And some patients have, this might be bad and this might be good, but this is bad. So it really is not like clear cut. But of course, the more you have, the better. I can tell you for sure, and it's pretty established in cytogenetics, if you have, there's 4 main chromosomes, 13q, 11, trisomy 12, or 17p. If you have 13q by itself, it's pretty good prognosis. But as soon as you add to that 17p, then your prognosis definitely goes down. So you can say, 13q is bad, but not really by itself. But if you add some other chromosome abnormality, like later on in your disease, you accumulate mutation, then it's not good. So of course, if you have ZAP70 and VH unmutated, and we've written some papers for at least these 2 markers, that's not very good, because if you have ZAP negative and unmutated, you're going to do better, because ZAP kind of trumps VH. And you can look at some papers there. So, it depends, and they're discordant cases. And it's a probability. It's not like, it's really always a statistic.
It's complicated.
It's complicated, unfortunately. We don't have yes or no answers to everything. And prognostic markers, okay, so I'm going to talk about the immunoglobulin VH heavy chain. Okay, this is a molecule like you saw on the slide. It's a transmembrane. It goes through the membrane. Some of it is sticking out it's in a receptor, an antigen binding receptor, and it has specific features that bind the antigen in the pocket of this molecule. And then these are generated by these mutations in the VH, which I'm going to explain to you, okay? And ZAP70 is another one, but I'm not talking about ZAP70 today. Now, the immunoglobulin molecules has basically is part of the B cell receptor. It's part of the B cell, and it's a receptor, so it's a receptor for the antigen. That's what that means. Now, the B cell receptor has 2 components. The number one is the recognition unit. This is the membrane, the immunoglobulin that binds to the antigen. So the first part that's sticking out. The second part of the receptor, the B-cell receptor, is the signaling component. That's like the arm that's sitting through the transmembrane molecule that's signaling to all the other protein inside the cells, and it turns on and off certain genes depending what signal the antigen gives to that first component of this molecule.
So is the immune globulin molecule the same as like the immune globulin that we measure in the blood?
That's secreted in IgM. It's like IgG, IgA. It's always, yes, they're related, but they're different from each other. Because that's why it's called IgG versus IgM. Because they're made doing different times during the B-cell development, and they have different functions. So, for example, I'll show you the picture. The immunoglobulin molecules, the IgM, IgG, IgA, they all pretty much look like this, okay? These kind of like V-shaped bits. But where they change is in this part of the molecule. So depending what sequence, this is C, it means constant region. That where usually there's not mutations in here, because this part doesn't see the antigen. This is cut underneath the cell, right? So these are like ones that are exposed. So in IgG is going to be different sequence here versus in IgM, IgA, and IgD, let's say. So they and each of them has the variable and the constant region, but it's particular to that isotype.
So this molecule, the immune globulin molecule, is on the surface of the CLL cell.
Yes.
So, but the immune globulin that we measure in the blood,
some of these get secreted. You measure by ELISA, right?
Right. So
in the plasma of the cell. So we can measure I'm talking about the ones that are stuck on the cell. And they're the ones that are causing all the immune reaction where the antigen binds and it trickles down the message to so on. So I'm not talking about soluble. I'm only talking about membrane bound.
Okay.
Because there's a whole bunch of other research on the soluble, how much you have.
Yeah.
But right now I can't get into it. But
No.
that's plasma. This is DNA from the gene that comes out and makes this molecule on the surface.
On the surface of the CLL.
Right. And we mostly, because it's CLL, we mostly use IgM. And now there's IgD on the surface, which is some stuff, but I don't talk about IgA and IgG and the other immunoglobulins.
Right.
Okay, so this molecule, let's just talk about IgM. I didn't even put that here because I just assume I always work with IgM. It's like, it has two parts. One is called the heavy chain, VH, and that's what you're going to hear me talking about. And it's heavy because you see it's longer, it's bigger, right? And this is a light chain. And why is it heavy? It's because when you do things that I can talk about, like you cut this molecule by enzyme, by trypsin, and you run them on a gel, this is a heavier band, so this is going to be higher up on the gel than the light band. Obviously, light kind of goes through a gel, electrophoresis gel, it goes down fast. A heavy band is bigger, and it's going to sit in a certain size. And so they call it, that's the heavy chain of the immunoglobulin, and the light chain. Now, most of our work that I'm going to talk about is the heavy chain, because they think maybe this is the more where the antigen, you see how it's made, the antigen or the unknown antigen, we are going to sit in here when the protein folds. It has more interactions with the antigen. There's a lot of research on the light chain, but I'm not really going to talk about it. And that is also gets very complicated. So, and you can see here the antigen binding sites are in this area of the molecule once it folds into a 3D structure. Now, like I said, the protein is made like this. This is a heavy chain where we look at the VH mutation status in this part. This part is a constant region, and it's constant because there's really not much going on in mutation-wise. It just identifies whether it's an IgM, IgG, IgA, and so on. So we don't really study. When we do PCR, we're looking at this little part of the molecule up here, because that's where all the action is occurring. We don't do PCR down here in the constant because it's going to be the same from every patient. And also, you can look at these, pretend this is like the framework of your house, like the walls. They don't really change. But what changes could be your shape of your window, that's going to make your house look prettier or uglier, whatever you want. But I'm just trying to make it simple. So this is where the things are going on. And this is the little antigen, which I'm going to go into in a minute. So, and you say, well, how do these molecules make? Well, what happens is, way back at the beginning of the B cell, the ontology of cell, It's called immunoglobulin variable diversity joining region rearrangement. And each of these makes up the heavy chain. It's just little pieces of genes that are all in your original DNA, in your germline DNA, that are kind of all lined up together like pearls on a necklace, like all little beads. And there is a bunch of them that have a particular sequence that are the heavy sequence. Then there's another pieces of DNA that are called the diversity region, and another little pieces of DNA that are called the joining of the heavy chain. And then there's a constant region, like I said, that really doesn't do much. It just kind of stays there. So what happens during rearrangement process, and this is random, one of these little pieces is going to get spliced by enzymes, is going to get stuck to a D diversity region, and it's going to get stuck to a joining region, and then it's going to eventually bind by ligases and things like that I can't go into, to a heavy chain. So if you follow this process, these get spliced and gets together. At the end, you've got a variable heavy chain, a little diversity region, and a JH joining region, which joins it to the heavy chain. Then there's transcription, RNA, and on and so forth. And at the end, you've got your sequence that we sequence by DNA, by PCR. The variable heavy is stuck to the constant heavy. And in between here, which is not, they're very small areas. They're only like 5 or 10 bases. ATCG is a GAH. It's very short. This is 200 base pairs. They're the D and the joining region, which is not marked. So that's what I'm talking about. And that's what comes in the protein above the cell surface. Now, diversity, I think, okay. So how can we have so many different VH genes? Like, how can you get diversity? First of all, there's the combinatorial diversity. This is a process of recombination from the vast diversity of the genes in the DNA. That allows a large number of potential combinations. Remember, there's many VH and many variable light chains. So any one of these VH can join with any other light chain, which is going to make a different molecule than another combination. So there's been some computational paper, then they said you can have almost millions of combination of what VH gene binds to what light chain, and therefore increases a lot of diversity in your B cells. Now, there is also junctional diversity, and those are the little D and JH regions that can come together, different ones, to make even more different molecule. And then above this, there's this mutation, somatic mutations, which is what can happen during the B cell development, which is what we see. This is kind of done by the time we get the cells. And then what happens during the lifetime of the person's B cell development, somatic mutation that added on. So, this normal B cell repertoire has found through evolution many ways to have a lot of variability in what kind of immunoglobulin you have on your B cell surface. And like I said, when we look at many CLL patients, we see that some patients use, like I said, there's many families. You can bind VH1 with VH or VH2. So some patients have the VH1 type of gene, some patient VH2, and so there's like basically really mostly 6 families that we identify in the VH sequences that we get from the database. So this would be like everybody that has red hair or blonde hair or light brown hair. People, they still have an immunoglobulin VH, but it's a little bit different than these guys because this sequence is ATGGC and this sequence is AGGGG or something in their DNA, and that makes a little bit different protein in the cell surface, and that will bind a different antigen. So there's a lot of variability, and we see that in the patients when we put all this data together. Sorry, this should be going forward. Okay, so what does the sequence look like? Like I said, it's just a genetic code, and each of these makes a codon, every 3 of them. So if a person is non-mutated, when we look at these, we line them all up to this sequence that's in the database. Let's say a person number 1, patient number 1, everything looks the same. You see there's no little changes. And you keep going at top line and everywhere, blah, 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 all the way down. That person does not have any mutation. He's 100% homologous to this germline sequence at the top line. This whole thing is about 282 base pairs.
But what is the germline?
Germline is the DNA that's like there when, like, before it's in your germline, like before when doing the ontology of the B cells. That's in the database, like in the world databases for, let's say, VH1 gene. This is what determines that VH1 gene looks like this. It's in the database. We get that from the database. So this patient, not knowing what the database in the world of VH research, we just match it and we see, does it match the VH1 sequence that's put in the data from the germline? Yes or no? Like this patient, it matches. These patients has 3% mutated, because if you go along, oh, look at the G changed to an A. So he's got a mutation. It's different. So these codon, it used to be AGG, let's say that was making proline and it was making a certain type of shape on the cell. Now it's got AAA. So that's isoleucine. It's a different amino acid. So this protein is already folded, and that's just one for example, it's folded differently than this amino acid because it's a different mutation right here. This is codes for a different amino acid to make a different protein. So if you go down, this person, the last person, we line him up. Oh, he's got a mutation here. He's got a mutation here. He's got a mutation here. You know, because it's different from this, like code, what do you call it? Like the germline.
Right.
That's our standard for that gene. And there, the G mutated to an A.
Right, but so why is it then that it's better to be mutated? It seems like it'd be worse.
Well, let me show you. Yeah, because, mutated, what does that mean? Okay, this shows the same picture again. Some patients are not mutated and some other ones, okay? So you get that. Now, why is it mutated? What happens if you change this shape, right? Now, if it's not mutated, this shape stays like this, and it can bind only this type of antigen, right? Okay. If this changes, okay, this can bind this antigen, or let's say another B cell there can bind this antigen, or it can bind this. You have variability in here. They can bind more than, you can bind different antigen depending which mutation you have.
So it's better for the cell to bind more antigen?
Because you don't want it to bind an autoantigen, which is the one that recognizes more the germline sequence. And autoantigens, let me go on, it's in the pictures. Yeah, the autoantigens are inside our like antigens in your body. So just to give you some background on antigens. Antigens are macromolecules that elicit an immune response. And they can be, like I showed you in the pocket. They can be protein, polysaccharides, or lipids. And they can enter the body from your environment, obviously, from outside sources you can inhale and ingest through cuts and everything else, right? An antigen can be generated, or they can be generated from the body, from your own body. They can protein from genes or virus that have infected the cell. They can have a aberrant protein made by cancer cells, or they can be autoantigens, okay? Now, they initially